What side of a zebrafish has the most stripes? The outside. Zebrafish are minnows from the freshwater ponds and streams of the Himalayan region in South Asia. They can be found in pet stores around the nation. Measuring 1.5 inches on average, these freshwater minnows make great household pets due to their resilience and iconic stripes. These are not like those fish, however. They have a special talent. They glow in the dark. These fish are just one example of the application of a process referred to as gene editing. Gene editing is an alteration of the base instructions inside the living cells of an organism. In this case, a special set of instructions was inserted into these fish to make them glow. These instructions are called DNA, the code of life, which makes up our genes. Every living organism has thousands of these genes, each one contributing to the unique features of that organism. Consequently, a wide variety of changes to an organism can be made simply by altering its DNA. For decades, scientists have researched ways to fix genetic errors through gene editing. Today, gene editing is easier than ever thanks to the CRISPR-Cas9 system. CRISPR can be used to make fish glow, detect pathogens, and even treat genetic diseases. CRISPR refers to the term clustered regularly interspaced short palindromic repeats, which are DNA sequences that provide the Cas9 protein with the instructions to precisely cut genetic material anywhere on the strand of DNA. These instructions from CRISPR contain guide RNA, which tells Cas9 where to cut, and another RNA that provides a structural foundation for the protein. Each target instruction requires a special sequence called a PAM sequence to bind Cas9 to the DNA, but this sequence can be found all over the DNA code, making the CRISPR-Cas9 gene editing system a very powerful tool to cut DNA. But why would we want to cut DNA? Cutting DNA lets us edit it. We can use CRISPR to edit genes in many different ways. We can use it to insert or delete genes, make small changes to the DNA sequence, or even detect diseases. Let's take a look at a few examples. These fish had a gene inserted to help scientists visualize parts of the fish to learn more about the process of development. These fish are transparent while developing, meaning that the glowing tissues shine through. Without gene editing, scientists would not be able to use the glow to see which parts of the fish are developing. CRISPR can also be used to remove a gene from the genetic code. Cutting the gene, or cutting around it, can make it stop functioning. This is called knocking out a gene. For example, this mechanism can be used to make a cell immune to specific forms of HIV. Removing the gene for one of the receptors that HIV uses to enter the cells with keeps it from infecting. By knocking out the gene that codes for the receptors for HIV, we can make a cell immune to the virus. This system can also be used to repair broken genes. One way this is possible has been shown with sickle cell anemia. Sickle cell anemia is caused by a change at just one point in the DNA, called a mutation, and mutations like this can be passed on from parent to child and affect families for generations. CRISPR provides an opportunity to change lives by fixing this single error, and has the potential to change the lives of hundreds of people. Foodborne illnesses, such as Salmonella, affect roughly 600 million people worldwide each year. CRISPR-Cas9 can detect pathogens with speed and accuracy. Scientists can engineer the guide RNA in the CRISPR-Cas9 system to recognize the presence of Salmonella in food. Once detected, a cascade reaction occurs that results in a readable color change, showing that Salmonella is present. Similar tests could improve health worldwide. CRISPR's capability to find and alter specific sequences has changed the world of science and medicine. Most of these applications are still in development, but the potential for growth is great. Just like our CRISPR fishies! We'd like to thank the following for their profound influence in the making of this video. Make sure to look in the description for the sources we used.
Which side of the zebra has the most stripes? The inside, of course. Except it's not that. Okay. Which side of a zebrafish? Which side of a zebrafish has the most stripes? The outside, of course.